Okay, so we're starting the material for Unit 4, which is going to be on your fourth exam. I have your exam scheduled for you. Do you remember when it is? The Monday of Thanksgiving week. Why am I torturing you by giving you a test the Monday before Thanksgiving? Because finals, basically. When you get back from Thanksgiving, you have your second lab practical to take. So if we put off this test, not only would it take up a whole class period for a test when you already have a major exam that week, but also it cuts into the time we have to cover genetics. Have y'all started genetics in lab yet? Yeah? No? Okay, today is, y'all are finishing, what was the last unit you did? The DNA fingerprinting, which is this exam stuff, okay? So you start genetics next week, and then you have two genetics lab, but one of them's online, I think, is the way that works, yeah. So you have 2 weeks of lab material on genetics. Genetics is new material for the final exam. We'll finish genetics, we'll start it and finish it the week we come back from Thanksgiving, and it's a major portion of your final exam. So I want to not rush through genetics. Technically, there are 2 chapters that are new material for your final, Chapter 13 and Chapter 15. The 15 is on technology, DNA technology. You're doing that in lab. I'm sprinkling it throughout, and you do have your review sheets posted. Okay, so now that we're getting into Exam 4 material, you do have reviews posted for this exam. It's just 2 chapters, Chapter 8 and Chapter 9. I keep thinking I want to add Chapter 10 in, and when we get to this point, the looks on your faces tell me 8 and 9's enough. Okay, so if you feel cheated about that, you can read Chapter 10 on your own. I'm just saying it's not going to be on your test. Is that all right with everybody? All right, as we get into DNA structure and function, we're going to be looking at kind of 2 lines of thought. I'm going to take this off because it bothers me. One is the history of not just who did it, but how they figured out what they did. And the other is what did they learn from that. So I've posted a number of assignments online for you in Canvas. And each of those assignments involves watching an animation. Most of the animations are 10 minutes or less. And then just writing a little paragraph about what the animation covered. Okay? Those all count on Test 4, all of them. The more of them you do, the more credit you'll get. So I want to strongly encourage you to hop onto Canvas, try to get some of those done. And the deadline to do them is by the time we sit down to take Test 4, that's the cutoff. You can't do them after the test. Everybody pretty clear on what to do in Canvas? Are we good with that? All right, as we get in, go ahead.
Is it going to be in the modules?
It's not in the modules, it's in the assignments.
Okay.
So when you can click on the link for assignments, or it should show up in your to-do list, although the due date's not until the Monday before Thanksgiving, so it may just not be showing up yet. So go to the assignments link and you'll see them for sure. Any other questions about that? Okay. So we're going to start with the discovery of DNA, and you are going to get tested on these people. So as biology majors, we kind of expect you to know who your forebears were. Okay, so can you name some of the people we've already learned? Anybody? Didn't we just learn one, about a week ago? Who figured out that cycle that we just love? That would be Hans Krebs. So we're adding some names. There are more names in this section because a lot of people worked on DNA fairly regularly and made some major, amazing discoveries. Okay, so the first one is Miescher. Miescher was German, and he worked in the 1860s. Look at the date of his discovery, 1869. He was a chemist. And as a chemist, he was looking for something to study that no one had studied yet. And it was suggested to him that we knew there were these things in cells called nuclei, but we didn't know the chemical makeup of nuclei. So a colleague of his suggested he work on what's in the nucleus of a cell. As a chemist, what's the chemical nature of it? And he said, well, you know what? Where can I get nuclei of cells in reasonably good quantities to actually study the chemistry? And they said, well, fish sperm have really big nuclei. And people raise fish commercially, so it's not hard to get fish sperm. When they spawn, the sperm just come out in huge numbers. So fish sperm, boy, that sounds really weird, doesn't it? But because they would raise fish commercially, it was something easy to come by. And then pus from bandages, that sounds really gross, doesn't it? But when you're working in a laboratory associated with a hospital, you can imagine a lot of that's available, first of all. But also they found that there's lots of white blood cells in that pus. Somebody had already put pus under the microscope to find out what it was and found out it's full of white blood cells, and white blood cells have really large nuclei. So those were two rich sources of nucleated cells for Miescher to work on. And he was able to figure out the technology. This is in the 1860s. He's figuring out the technology to isolate just the nucleus of the cell and then chemically analyze it. So he gets to name it, right? So what did he name it? Nuclein. That's a catchy name, I suppose. Seems obvious, doesn't it? But once it was determined it was an acid, what did we call it after that? What's DNA? What does it stand for? Deoxyribonucleic acid. So that's where the term nucleic acid came from. Miescher named it nuclein and then developed the techniques to study it. All right, so we, from the 1860s, know there's this stuff called DNA that's found in the nucleus of all nucleated cells. And look at how much later we are now. We're in 1928, and Frederick Griffith did a very important work. One of the links I put up in Canvas for you is an animation of Griffith's experiment. It was a very elegant experiment, and appreciating the experiments that these guys did will help you appreciate the field you're going into and understand the results they got. Griffith worked with bacteria. He didn't just work with any bacteria, he worked with strep. And he didn't work with just any strep, he worked with not the thing that causes strep throat, but its cousin, the thing that causes pneumonia, Streptococcus pneumonia. And strep pneumonia is very often fatal, even today. It's fatal sometimes in people. He didn't experiment on people. He used lab mice for his experimental animals. But what he demonstrated was a phenomenon that he called bacterial transformation. And transformation is a phenomenon in which bacteria can pick up something from a different strain that turns them into a virulent form. Now, what does virulence mean? What's the difference between a virulent bacteria and a non-virulent bacteria? Anybody? Use that term in common language. It's the same word root as going viral. Okay? So a virulent strain is one that can spread and cause disease. Makes you sick, makes you die. Okay? So which one would you rather be infected with, a virulent strain or a non-virulent strain? Let's go with a non-virulent if we have the choice, okay? But transformation occurs when you take a strain that's non-virulent and you pass to it something from a strain that is virulent, and it transforms into a virulent strain. And Griffith played with this. He discovered it, and then he started experimenting on it to see what on earth could it be that's causing this transformation. What is it about a virulent strain that makes it virulent? All right, so we're going to take just a brief look at some of his experiments, and then I'm going to send you online later to watch the animation. Are you with me? All right, so he worked with two strains of bacteria. The virulent strain was called the S, and the non-virulent strain was called the R, okay? They were called this because when the bacteria were grown on petri dishes in colonies, the R strain, the colonies had a rough outline to them. They had a rough edge to them, and the S strains were smooth. So it's the same R and S as in rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay, so far? So it was just from the appearance of the colonies, but it turns out that if you inject mice with the R strain, it's non-virulent. And if you inject mice with the S strain, it is virulent. It kills the mice. Are you with me so far? So there's something about the S strain. It has something that makes it kill mice, and that's something that the R strain doesn't have. Okay, good? Got it? Which one's virulent? R or S? Which one's virulent? S. How do we know that from reading the screen? If you inject it with S, the mice died. Okay? So what if you kill the S strain bacteria first? You just run them up to high heat, you know they're dead. How would you know they're dead? Now, you're working in a microbiology lab. You're growing them in a broth. You're putting them on plates. They grow into colonies. So if I put that little broth with live bacteria in it, but I put them at high heat, and then I put them on a Petri dish, what happens? If they're all dead, nothing grows, right? So he knows for sure he's injecting the mice with dead S. Live S kills the mice. What about dead S? What does that do? Can you read the screen? They live. So that proves the bacteria have to be alive to cause the pneumonia that kills the mice. Are you with me so far? So if you have live R, that doesn't cause pneumonia. If you have dead S, that doesn't cause pneumonia. The only thing that causes pneumonia is live S, right? So what he did next was the part that nobody else had ever thought of. He thought, what would happen if I mix dead S with live R? What would that do? What would you predict it's going to do? The S are dead. They can't reproduce. You cannot make any more S. The R bacteria are alive, but they don't make the mice sick. So, but when he mixed them together and injected them in the mice, what happened? So dead S plus live R, neither one individually will kill the mice, but when you mix them together, what happens? Okay, so first of all, the mice died, which they shouldn't have done, right? And second of all, when he isolated bacteria from the dead mouse, it was live bacteria, but it wasn't R anymore. It was S. That's pretty wild. So this is why he called it transformation. What happened to the R bacteria? They got transformed into S bacteria. Okay, does everybody understand the profound importance of what Griffith was able to do? That is our first clue that something that you can isolate from bacteria could be figured out to be what causes the genetic makeup or what causes the characteristics of an organism. All right, we all know there's something we inherit. Griffith is the first one to be on the track to figure out what that something is. All right, questions about that? All right, so here in the diagram, this is the diagram out of your book. You need to be able to answer questions about this on the test. So what about live R? What does it do? Look at figure A. Mouse is running around, no symptoms. What about live S? That's figure B. That's kind of sad the way they drew it. Usually we just draw little X's for their eyes. Okay. Dead S is figure C. What do you see? Mouse is still fine running around, but what about mixing live R and dead S? Not only did the mouse die, but what did we isolate in the mouse's body? S, when we didn't inject them with live S. So somehow those living R's got transformed into S. They picked up something from the dead S cells they were mixed with, and whatever that something was is the transforming principle. And now all we have to do is track down what that is. Okay, so far? Questions at this point? All right, so the question is then, what is the transforming principle? What is it about the dead S that transformed the live R? So we're going to move on to 1940. How much time has gone by? When did Griffith do his work? Did y'all write it down? I don't test you on dates per se, but you have to have some kind of frame of reference. So when did Miescher first discover nuclein? 1869. And when did Griffith do his work? 1928. So now we're jumping to 1940. Why do you think we made such a big jump from 1928 to 1940? There was a little bit of global upheaval in the meantime, like, depression and global war and little things like that. Probably got in the way. Okay? So Avery, who worked with some colleagues, took the first steps to determine what that hereditary material is. He repeated Griffith's experiments first. First, he did it over again. This is kind of classic in science. If you have something earth-shattering, other people ought to be able to repeat your experiments and get the same results. So Avery did that. And then with his colleague McCarty, what they did was they took those dead S cells and they divided them into their chemical components. What are the 3 chemical components they divided them into? Read the screen. Lipids, proteins, and thanks to Misher, we know what nucleic acids are, and we can chemically analyze them. Are you with me so far? So being able to isolate the lipids from the proteins from the nucleic acids, what he could do then was take just the lipids from the dead S, or just the proteins from the dead S, or just the nucleic acids from the dead S, and mix them with the live R, and see which ones were transformed. Are you with me on that? So you and I already know the answer. What was the transforming principle? It was the nucleic acid, right? So when he mixed the lipids from the dead S with the live R, mice didn't get sick, our cells were not transformed. When he did the same with the protein, by the way, at that time, everybody was pretty sure protein was it, that protein was the heritable material. Nobody had a clue that it was the DNA. Everybody thought it was the proteins. So everybody was surprised when Avery and McCarty came up with nucleic acid being the transforming principle. And they made them do it over again. And then they made them do it over again. And then they devised other experiments to show yet again that it's not the protein. Everybody was sure it was the protein, but it's not. It's the nucleic acid. And he was even able to, in his repeat experiments, keep just the DNA and get rid of the RNA and show that it's just the DNA that transforms the R bacteria into virulent S bacteria. Okay, so he used Griffith's experiments as a starting point, redid those experiments with just these extracts from the dead S's, and showed definitively DNA is the transforming material. Are you with me on that? So from Misher, we know what DNA is. From Griffith, we kind of learned that something can turn one kind of bacterium into another kind. And now from Avery and McCarty, we know what that stuff is. It's the DNA. Okay? Now put yourself in 1940. And when you think about what we knew at that time, I mean, we barely had antibiotics invented by then. So the state of our knowledge of cells and biology was pretty primitive at the time. So this was a big deal. All right, so not that long after, in the 1950s, Hershey and Chase did something first. They confirmed that Avery and McCarty were right, because a lot of people doubted it. A lot of people thought protein was going to be it. And then they did something even cooler, just to latch down that, yes, it's the DNA and not the protein. They worked with bacteriophages. Bacteriophages are viruses that infect bacteria. They're really big, they're easy to cultivate, and they're easy to look at under the microscope. All right? So what they were able to do was, again, to separate the protein from the DNA in the virus and infect the bacteria with one or the other and show which one infected the cell. Let's see if we have a picture of that. There's Hershey and Chase. Hershey was one of the few women who was actually recognized for her work at this time. Most of the time, women like Rosalind Franklin were kind of in the shadows. And in fact, she started out as a graduate student and probably had to, fight to get recognition. All right, look at this figure. It looks like some kind of lunar landing module, doesn't it? That's actually what that virus looks like. And on the right, that's actually a microscope view. That's an electron micrograph of the actual virus particles infecting bacterial cell. Okay? So this is really a wild and crazy looking virus. The entire outside of the virus is protein. It has a protein coat, even those little tail fibers. That's all protein. And the DNA is on the inside. Now look carefully at the figure on the right. Do you see the little green lines coming down into the bacteria? Can you see that? Okay, so from this view, now this is not what Hershey and Chase did. This is much more modern than that. But what we can see in this view is it's injecting something into the cell. What Hershey and Chase knew, not from pictures as pretty as this, was that entire thing that looks like an artificial machine doesn't enter the cell, that only a very small part of the virus enters the cell to infect the cell. And the question they wanted to answer was, is that stuff that gets injected, is it protein or is it DNA? Because they're going to prove or disprove what Avery said, which was that the DNA was the transforming principle. So this is the experiment that's just going to latch down on that. So what we see here is that what Hershey and Chase did was that they labeled with radioactive sulfur just the protein. Now, I want you to stop and think. We learned 20 amino acids. At the time, I told you 2 of those have sulfur in them. Do y'all remember that? Just lie to me and say yeah. Because at the time, I told you this is going to be important later. Well, this is later. Amino acids have sulfur. Just 2 of them, but some amino acids have sulfur. Okay? What is a nucleotide made of? Do y'all remember what that's made of? Nucleotides are adenine, guanine, cytosine, thymine. They're nitrogen bases, so they have nitrogen, carbon, hydrogen, and oxygen. There's nothing unusual there because you find that in amino acids as well. But they also have a phosphate. Is there any phosphate in protein? None. But there's phosphate in DNA. So what Hershey and Chase figured out is that they could use radioactive labeling of sulfur to tag the protein and of phosphorus to tag DNA. So first thing they had to do was grow bacteria with a supply of radioactive sulfur, infect it with bacteriophages, let the bacteriophages grow up, and since they've been grown in radioactive sulfur, which part of the bacteriophage is radioactive, the protein or the DNA? Which one has sulfur? The protein does. So if we grow the bacteriophages in radioactive sulfur, we're marking the protein. So they grow the bacteriophages in radioactive sulfur, and then they transport them to a brand new, no radioactivity bacterial colony. So the only thing that's radioactive is the bacteriophages. I'm going to take these radioactive bacteriophages and inoculate some bacteria that I've got in a new colony. Are you with me so far? So that's what we see in the picture. So where is the radioactivity in that picture? It's pretty tiny up there. It's the red, okay? So it's just in the coat. So when the bacteriophage injects its hereditary material, its transforming material into the cell, what they would do is give it just enough time to inject its stuff into the bacterial cells, and then they would stick it in a centrifuge, and that would break the protein coats off the outside. And they would separate the bacteria from the broth. Where are the protein coats? Are they in the bacteria or are they in the broth? Did the protein coat get injected into the bacteria? No. So they shook it loose. They ran it through a centrifuge. The bacteria are in the little pellet at the bottom of the centrifuge. The protein coats are all in the broth. Which one's radioactive? The broth or the bacteria? The broth is radioactive. So we know that the protein coat kept the radioactivity. So we know that it had amino acids. Now, part 2, what are we doing here? What are we doing in our bottom picture? Now we're using radioactive phosphorus. Where does the phosphorus go? Into the DNA. So I've grown my bacteriophage colony in radioactively labeled phosphorus. I take those and put them in a new batch of bacteria, run the experiment again, but this time where's the radioactivity? It's in the cells, okay? So from this 2-step experiment, Hershey and Chase demonstrated that the protein does not enter the cell, only the DNA enters the cell, okay? So who's the transforming material? It's got to be the DNA. So this was in the 1950s. Does that seem like a long time ago to you? It does to you. Trust me, I know. But let me tell you, from a biological perspective, that's not that long. 60 years, more or less, 60-something? Yeah, it seems like a long time. We've written a few textbooks between then and now. So the Hershey-Chase experiments definitively show that protein is not the hereditary material, that it's the DNA. Okay? Can you explain that experiment? And can you remember who did it and about what time frame it was? In the 1950s, Hershey and Chase. Okay? There is online an animation of that you can earn a little credit for writing a summary of. That's one of the assignments I posted. So you have one for Griffith, you have one for Avery, and you have one for Hershey and Chase that you can watch just to refresh your memory of these things. Okay? They're parked in Canvas for you. All right, so now we know DNA is something we're interested in, and the reason we're interested in it is we know it's the hereditary material, and we would like to understand how on earth something that simple could be the basis of all the variety of life on earth. So we're going to look into the structure of DNA. We're going to chase down the discovery of DNA structure. You and I already know that nucleotides are the building blocks. Through the work of a lot of chemists over the years, this much we already knew. We have, in DNA, we have the 5-carbon sugar deoxyribose. We have at least 1 phosphate group, up to 3, right? ATP has 3 phosphate groups. And then 1 of the 4 nitrogen bases. Do you remember what the nitrogen bases are? Go back to your notes from chapter, I think it was chapter 2 or 3 when we did this, okay? It was test 1. I did it then because I knew I had to come back to it, and I knew when I came back we'd be in a hurry, okay? So you have 2 purines and 2 pyrimidines. The purines are adenine and guanine, and the pyrimidines are thymine and cytosine. That's in DNA. Not worried right now about RNA. We're focused just on DNA. Okay? So that's just a quick review of what we already knew. Do you need to be able to recognize these on tests? What did I tell you before? Does anybody remember what I made you know for test 2? I made you learn the difference between a purine and a pyrimidine. Okay? I will not expect you to tell which one's adenine and which one's guanine. But how do you tell a purine from a pyrimidine? Purines are on the left in this figure. The pyrimidines are on the right. Look at what's colored blue. So purines have 2 rings, right? Pyrimidines have only 1. Okay? Which one has the longer name? The one that's the shorter structure. Okay? Bigger structure, shorter name. All right? You need to know purines are adenine and guanine, and you need to know purines have a double ring. Okay? You need to know the pyrimidines are cytosine and thymine, and that they have a single ring. All right? Now that's not all. That's just to be able to recognize them from a diagram. Okay, so now we're going to move on to the next guy that there's a video online for you to do as an extra assignment, Erwin Chargaff. And Chargaff actually died not that long ago, only about 15 years ago, but he did his work mostly in the 50s and 60s. And what he figured out is that, and he's a chemist, he's not a biologist, that it didn't matter what organism it was, whether it was a bacteria or a cabbage or a rabbit. It flat doesn't matter. That in all cases, if you totaled up the concentration of adenine, guanine, cytosine, and thymine, that there was always a rule that they followed. However much adenine there was the same amount of thymine. And however much guanine there was the same amount of cytosine. Now, on a test, I should be able to ask you to figure that out, okay? We'll do it from percentages. So the percentage has to add up to 100%. Doesn't matter if you're an amoeba with, 80 chromosomes or a human with 46 chromosomes. Whatever the amount of DNA you have adds up to 100%. Are you with me on that? Okay, so if, of that total, however much adenine there is, there's going to be the same amount of thymine. Okay? So if I tell you there is 10% adenine, what's the percentage of thymine? 10%. What's the percentage of guanine? Can't be 90, because I'm 10% adenine and 10% thymine. So how much is left? 80% total, of which half is guanine and half is cytosine, right? So you could figure that out. Well, that's basically what Chargaff was able to figure out. Doesn't sound all that mind-boggling, but nobody at first knew what to do with that. They just knew it had something to do with how these molecules were built, and they weren't exactly sure what to make of it. What they did know was that it's species specific how much adenine you have, and therefore how much guanine you have. If you know one, don't you know the other three? Right? So if guanine is 35%, then what? Cytosine is what? 35%. So that's a total of 70, leaving you with 30% to split between adenine and thymine. So as long as you know any one of those four, you know the other three, and every species was unique. Okay, or at least to the level of genuses, it's unique. So if you look at a cabbage plant versus a fish versus a human versus a bacterium, they all follow Chargaff's rules, but their own unique combination of purines and pyrimidines was species specific. So something in that will help us figure out what the structure of DNA is. Any questions so far? So what are Chargaff's 2 rules? The first rule is A equals T and G equals C. What's the second rule? Differs among species. Every species has a unique percentage. All right, so now we're going to leapfrog to Wilkinson, Franklin, and Watson and Crick. And well, I've already told you this story once. It's one of my favorite stories. It's one of the most legendary stories in modern biology. Watson and Crick published their paper in 1953, just so you have some kind of frame of reference. And you might remember that Watson and Crick weren't really supposed to be working on DNA, but being young researchers, they were trying to find something interesting to work on. And they knew that Linus Pauling had just published the structure of protein, the alpha helix structure of proteins. And that gave us a hint that there was probably a helix in there somewhere, or could possibly be a helix. They also knew that Pauling was working on the structure of DNA. Now, Linus Pauling was working in the United States, somewhere in California. These guys were in London, or somewhere in England. And Wilkins and Franklin were working in London. And Wilkins was working on the chemistry of DNA, and Franklin, remember, was working on what is the three-dimensional structure of DNA. And you might recall I told you that Wilkins thought that Franklin was his technician, and that's not what she thought. She already had a PhD. She had her own lab. But they were both working on DNA. So when Watson and Crick found out that they were not being allowed to work on DNA, all that meant was they were not going to tell anybody they were working on it. But they found out that Wilkins was, and they went to his lab and befriended him. And Wilkins was like, oh, you want to look at it? So Franklin had already presented her results. She just hadn't finished nailing them down and publishing a paper yet. But she had shown her very famous photograph, which is at the bottom of the screen. And what you're able to discern from that photograph, if you know what you're looking at, is that it's a double helix. You can tell it from the photograph. Okay? If it were a single helix, what you would see that looks like an X would only be one line, one diagonal line, not two. And the fact that that X is divided into little dashes just shows you that it's spiraling. Okay, so if you think of a spiral staircase, and if you were looking down on it, you could see the steps individually, that's what those little dashed lines would represent. Okay, so this is her original research that she hasn't published yet. And what Watson and Crick want is to get the measurements off the photo so that they know exactly what the bond angles and what the size of the molecules needs to be. And so she wasn't in the office, and so Wilkins let them into her lab and gave them her photograph, which we don't do that. Just so you know, we don't go take somebody else's research and take measurements off of it. But they were racing. Why were they racing? Because Linus Pauling was going to beat them to it. Now, Pauling came out with his own proposal of what the structure of DNA was, but by the time he did, Watson and Crick already knew he was wrong. Because I think he proposed a triple helix, and they already had access to this photograph, so they knew it was a double helix, and they knew he was wrong, but that just made them want to go even faster to try to figure it out. There is a video online that actually shows, it's an interview with Watson, and it shows the structure as they built it. So I highly recommend that you watch that one. If you're going to watch it, you might as well earn some extra credit in the process. So Watson and Crick were able to figure out that there are two nucleotide chains, they call them strands, running in opposite direction and coiled into a double helix. The whole idea of a double helix was based on Franklin's photograph. But Watson and Crick did the work of figuring out the chemical angles, the molecular structures, and how it would actually work. She wouldn't have done that. That was not her field. Her field was X-ray crystallography. So why didn't she get a Nobel Prize and they did? She died before the prize was awarded. A Nobel Prize is not awarded to people after they die. You have to be alive.
Would she have gotten it?
Huh?
Would she have gotten it with that?
The problem with giving it to her without that is the Nobel Prize is never awarded to more than 3 people. So since there were 4 of them distinctly involved, they probably would have just passed them over and awarded the prize to somebody else for something different. That's just the politics of the Nobel Prize. So if we go back and look at this picture again, Watson and Crick in this picture, that's Watson on the left and Crick on the right, built this model themselves. Actually, they had the guys in the machine shop build their little nucleotides for them. So you remember those pictures we looked at of uracil, or not uracil, of the bases, this picture? They drew those and cut them out of cardboard with all the bond angles exactly right. And then they gave them to the guys in the machine shop and said, can you make this out of metal plates for us? And they just made a bunch of copies of it, which they then assembled into their model. And so they showed that the bond angles were exactly right. And that's a phenomenal trait. I'm not saying they didn't deserve the Nobel Prize. I'm not saying that. I just can't get over the fact that Franklin got cheated. She got cheated. Plus she died of renal cancer. Why did she die of renal cancer? Probably from working with the radioactive elements. Probably. Y'all remember that when you picked what field you're going to go into. Okay? All right, so when we look at this structure, this is what we know based on what Franklin did. Can you read those numbers up there? 0.34 nanometers between each base pair, a 2 nanometer diameter, and a 3.4 nanometer length of each full twist of the double helix. I do kind of want you to know the numbers, but it's intriguing to me that it's exactly 10, a magnitude of 10 difference between each base pair and between one twist. Okay, so it's 0.34 nanometer between each base pair and 3.4 nanometer length of one twist. So that's 10 times. That means there's 10 base pairs in one turn of the helix. The importance of these numbers is not that much that you remember what the numbers are, but more that Watson and Crick could measure what those numbers would be when they built their model. So when they built the model, they knew that it agreed with these numbers. Where did these numbers come from? Who gave us those numbers? Rosalind Franklin did, off her photograph. Okay? So she was able to derive these distances based on analyzing the X-ray crystallography. Okay, so that's the structure of DNA. Now, when it comes to the difference between bacterial chromosomes and eukaryotic chromosomes, boy, that's a big difference. It's a huge difference. When we study in 1407 the diversity of life on Earth, we spend a little more time talking about bacterial chromosomes and eukaryotic chromosomes. One of the main differences I think you should know between them is that a bacterial chromosome is circular, whereas eukaryotic chromosomes are linear. You knew our chromosomes are linear, right? Okay, but bacterial chromosomes are circular. Archaeal chromosomes are usually circular. So what's in a chromosome besides DNA? It turns out there's a lot of different things. And one of the most important things in a chromosome is there's a lot of protein. And the proteins that are found in the DNA help keep it organized. They keep it packed. They regulate when a gene is turned on or turned off. They even regulate generational changes. So there are some genes in your DNA right now that were first turned off maybe in your great-grandparent. And once they got turned off 3 generations ago, they've stayed turned off this whole time. And we don't know a lot about that regulatory level, but we know it's due to the proteins or the enzymes that are involved. So, of course, we know that a chromosome is, the most important part of the chromosome is the DNA, and it's the DNA that carries the cell's genetic material. The problem that everybody had, the reason everybody thought it had to be the protein and not the DNA, is that proteins are more complicated, and we're complicated things. So surely it needs to be something complex. How many different amino acids do we use to build proteins? How many? Anybody remember? How many different amino acids do we use to build proteins? 20 of them, right. How many different nucleotides are there? Adenine's one. What are the others? Guanine, cytosine, thymine. There's only 4 of them. So how can 4 simple things like that possibly be complicated enough to create all the genetic diversity in life on Earth? Well, of course, we figured that answer out eventually. All right, so chromosomes, we already know from our study of cell division, are only going to appear during mitosis, right? We pack them into visible chromosomes only during mitosis, but by the time we do that, we've already replicated the DNA. Now, when we studied this before, we looked at karyotypes, remember? And we studied mitosis, cell division, and you have to send one of the 2 identical copies to each daughter cell. Now we're going to study how is that process carried out. Okay, how exactly do cells replicate their DNA? So this is just kind of a review. You remember studying this in lab when you did mitosis, that you have a centromere and chromatids and a duplicated chromosome. And we have these different proteins. One of the most important proteins in our chromosomes is a group of proteins called histones. Histones are basic proteins that they form up in little clusters of 8, and the DNA coils around them. So they're like little spools that we're going to use to spool the thread of our DNA. And so each little spool is called a nucleosome, and then we're going to coil up our nucleosomes into even more complex structures. So what's a histone? It's the protein that you make the spool out of. Okay, what's the nucleosome? What's in the nucleosome? Does it have DNA in it? The nucleosome is the DNA wound around the histones. Okay? So an empty spool and some thread. The spool with thread on it. So what's the thread? The thread is the DNA. The spool is the histone. The spool with a thread on it is the nucleosome. Okay? So that's the basic organizational plan of our chromosomes. Bacteria don't do that. Bacteria don't even have histones. They have a totally different set of proteins to organize their DNA. So in this picture, can you all see that well enough? That's really tiny. This is the picture out of your book, but you're going to take our nucleosomes. That's number 2 on our slide. Let me see if I can get this to work. Yeah, my pen's not working for some reason. All right, so right in here, you can see a little cluster of nucleosomes. What's happening here? I'm taking my nucleosomes and I'm kind of coiling them up. We call that supercoiling. And then what do? I take those coils and coil them up, and then I take those coils and coil them up. How many layers of coiling do I have here? It's like coils on top of coils on top of coils. How condensed is the DNA in a chromosome? It's crazy condensed.
So the histone is what the DNA is wrapped around?
Yes, the histones are the proteins that form the little spool that we wrap the DNA around. Okay, so here's some images just showing you how coiled up it can get. So on the far right, you see a duplicated chromosome, and you can tell that it's coiled up, little threads. All right, how many chromosomes do we have? Total. 46. How many different kinds of chromosomes? 23 unless you're a guy, right? Then you have both an X and a Y, whereas us females, we have 2 X's. So I have 23 kinds of chromosomes. You guys have technically 24, okay, because you have both of the sex chromosomes. What does the 2N stand for? See that on the slide? 2N. Did y'all learn that term in lab? You will when we get to genetics. 2N means, we call it diploid. It means you're a cell that has all 46 chromosomes. Our sex cells are only N. We call that haploid. So in a sex cell, a sperm cell or an egg cell, you only have 1 copy of each of the kinds of chromosomes. Now, we already did karyotypes, right? Y'all remember doing this? So is this a karyotype of a diploid cell or a haploid cell? How many copies of each kind of chromosome do you see? 2 of them, right? So that's diploid. Is this a male or a female? Look at that picture carefully. Male or female? Female, how do you know? It's got 2 X's and no Y's. Okay? All right, so if it's a sex chromosome, in humans it's the X chromosome or the Y chromosome. Other animals have totally different arrangements. In some animals, some insects, if they have a sex chromosome, it's a female. If they don't have a sex chromosome at all, it's a male. And other animals and plants just do it totally differently. So if it's not a sex chromosome, if it's chromosomes number 1 through 22, we call those the autosomes. All right? That doesn't mean that they don't have anything to do with sexual reproduction. It just means that they're not the sex-determining chromosome. So there's something on the X chromosome and there's something on the Y chromosome that determines sex. And in fact, it's on the Y chromosome, and it's a gene that turns on testosterone production. So if you don't have testosterone production prenatally, like within the first few weeks after the egg is fertilized, then you're kind of going to be a female. Okay? You've got to turn on that testosterone gene. If it's defective, if it's not working right, or if the receptor for testosterone is mutated and doesn't work right, you can have a Y chromosome in every cell in your body and still look female. Because there's a gene on the Y chromosome that when it's working normally determines the male sex. So, you know, we're used to thinking that sex is either, you got 2 choices, male or female. And I'm going to tell you it's a lot more complicated than that. There are some instances where that little gene that's supposed to be on the Y chromosome has accidentally transferred to the X chromosome. What does that do? You'd be a male with 2 X chromosomes. If the sex-determining gene migrated from the Y chromosome to the X chromosome, you'd be XX, but you wouldn't know it unless somebody checked your chromosomes because you would be a male. Okay? So is sex really that simple? You're either born a boy or a girl and that's it? Yeah, it's not that simple. Sometimes babies are born with indeterminate sex organs. They didn't develop properly, and you really can't tell at birth whether they're a boy or a girl. So anybody that says it's that simple, XX is a female and XY is a male, doesn't know enough about the subject to have an opinion, in my opinion. Okay? All right. We are going to start on, what's today? Wednesday? Is it Wednesday? Yeah. So you know I won't be here the rest of this week. So on Monday, we're going to start with DNA replication. We are not finished with Chapter 8. We're going to finish 8 on Monday. We're going to work on 9 Wednesday and Friday. And then we're going to take our test the Monday after that. Does that sound exciting and wonderful? And I know you're thrilled. So y'all have a nice long weekend. What else do you have to do between now and Monday? You better finish that research paper. Don't forget. All right? See you next week.